The initial outbreak of the Equine Influenza virus, namely EIV, over 15 years ago worldwide, caught many horse owners by surprise. Recently, the ability for the influenza type A virus to cross the species barrier from horses to dogs has sparked much scientific investigation and has led to a major human health concern. In a 2003 research study, Hughes et al. Examined the transmission of EIV during an outbreak in the town of Newmarket, United Kingdom. Newmarket has a high thoroughbred horse population and has many established horse training properties. Although vaccinations for EIV is mandatory for racehorses in the UK, the town experienced a large epidemic of the virus during this time. The research aimed to investigate the transmission of EIV from horse to horse throughout the town. The study also aimed to better understand the intra-host genetic variations between horses contracted with the EIV virus. Nasal swabs were collected from 120 horses in various Newmarket stables over an 8-week period, as Figure 1 illustrates. Swabs were analysed and transmission mutation networks were detected, and a phylogenetic tree was formulated. Then transmission networks were examined, and chains of horse connections between horse establishments were reconstructed. These reconstructions allowed Hughes and his team to focus on how EIV genetically diversifies and is transmitted between horses. Hughes found that similar to other RNA viruses, EIV intra-host population and mutation rates were high, with viral variants exchanged during transmission. This evidence suggested that loose transmission bottlenecks allow the multiple passages of variations between horses. The graph in Figure 4a showed the highest number of horses infected with EIV was 58 horses on April the 9th. Figure 4c also indicates the reproductive nature of EIV, with secondary cases indicated by the black dots. Hughes and his research team found that the high viral genetic turnover could have been caused by environmental factors such as humidity, temperature, and horse immune system status. The study concluded through combining epidemiological and viral sequencing data to prove that high contacts between EIV infected horses allowed the virus to therefore become stronger and a higher chance for an epidemic to occur. EIV can therefore be controlled and isolated to specific horse establishments through vaccination programs.